Let's learn biology with Mandewi. Believe me, biology is fun. Okay, so today we want the detection of starch in food sample, and the objective is to test for the presence of starch in a given food sample. So this is the theory for that.
And today we're talking about carbohydrates. Carbohydrates are a macromolecule made up of carbon, hydrogen, and oxygen in a 1:2:1 ratio. So there is 1 carbon for every 2 hydrogens and 1 oxygen in all carbohydrates. They are also known as sugars and have several key functions in living organisms: rapid energy production through glycolysis, energy storage as glycogen in animals and starch in plants, and a structural component of plants as cellulose and crustaceans and insects as chitin. The simplest form of carbohydrates are called monosaccharides. Mono means one, and saccharide means sugar, so a monosaccharide is a single carbohydrate molecule. Examples include glucose, an important simple sugar that is a key energy source in living organisms and the core of many structural carbohydrates. Fructose is a sugar found in many fruits. Galactose is a sugar found in yogurt and is less sweet than fructose and glucose. And ribose is a monosaccharide that helps form DNA. You are probably familiar with some disaccharides, which are 2 monosaccharides chemically bound together. Examples include sucrose, which is common table sugar and is formed from glucose and fructose, and lactose is the sweetener in milk formed from galactose and glucose. People who are lactose intolerant have low levels of the protein lactase, which breaks down lactose, and if they can't break down lactose, then the stomach will get pretty upset. When more than 2 monosaccharides are combined, a polysaccharide is formed. Starch, glycogen, and cellulose are all examples of polysaccharides, and they're all made of the same monomer, glucose. The way glucose is bound together determines whether the resulting saccharide is starch, glycogen, or cellulose. Needless to say, carbohydrates are pretty important.
Conclusion is carbohydrate is divided into polysaccharides, oligosaccharides, disaccharides, and the simple one, monosaccharides. Okay, so the indicator is iodine solution. If the food sample given by iodine solution, and the color will change into blue-black color. So this is the procedure before our virtual lab.
Materials required: test tube, starch solution, that is, water extract from boiled rice, iodine solution, dropper, test tube holder. Procedure: take a few milliliters of starch solution in a test tube with the help of a dropper. Now, add a few drops of iodine solution to it. Observe the change in color of the mixture. Observation: the color of the mixture changes to blue-black.
So let's start our virtual lab about the start test. For the first one, please open this link. For the first one, we choose the food sample and then drop with 1 or 2 iodine solution, and after that look at the color change and do the same with the next food sample. Okay, after all sample tested by iodine solution and you note all color changes, and then screenshot your screen, upload to Scholar, and don't forget to write your conclusion.